Good evening to all of you. I am Dr. Avat Shah. I am welcoming you all on the behalf of Bionivid into our ongoing interaction service into the research verses. Today we have with us Dr. Prashanth, who is a senior scientist at Department of Biotechnology and Bioinformatics at Birla Institute of Scientific Research. I know Dr. Prashanth from last 3 to 4 years. We have been working uh, with him since then, and he is a very great person. He is having a very great personality, very humble person. I'm sure you all will going to enjoy his presentations today. Now, to introduce Prashant sir, he is an experienced researcher in functional and medical genomics. He's also having some good knowledge about protein informatics and system biology also. His specialized interest is in extracting biological knowledge in the domain of next generation sequencing. He has strong inclinations for publication and thriving towards academic excellence, teaching and raising collaborations. So this is a short introduction for Dr. Prashant. I'm handing over to you, Avinash.
Hello. Welcome, Dr. Prashanth. Welcome, everyone. My name is Avinash, and I'll be moderating today's session. So I have a small request to make. All of you are requested to kindly drop in. I'm sure during the course of the presentation, all of you must have some queries regarding the topic that is being presented. So rather than disturbing the flow of Dr. Prashanth in the middle, I would request all of you to just drop in all your queries in form of private messages to me. I'll accumulate them one by one and then convey the same to Dr. Prashanth after the end of his presentation. I hope, first of all of us are audible to you. You were able to hear all of us properly. If yes, then kindly drop a yes in the chat section. Okay, great. So I would request all of you to drop your queries as a private message to me. I'll keep a note of them and convey them to Dr. Prashanth as soon as his talk ends. Okay, so that we can have a discussion post the presentation as well, and his flow doesn't get disturbed, right? So over to you, Dr. Prashanth.
Yes, thank you so much, Avinash and Avat, for your kind introduction. I hope I'm audible.
Yeah,
Yes,
please
sir,
drop
you are.
yes or no in the chat section, everyone. I hope you're able to
Yes.
hear Dr. Prashanth.
Yes, I think, yeah.
Yeah.
So I'm just, going to start my full screen. Yeah. I hope everyone could see the screen, Avat and Avinash. Yeah.
Yes. Yes, sir, the screen is visible properly.
Right. Yeah. Yeah. So first of all, thank you so much, for this great honor, Avat and Avinash and Rohit. In fact, Bionivid is doing great guns the last several years, and I've known Avat, for over 3.5 years, ever since he was in Excel race. And
Yes.
a part of the library preparation that we have done for the talk that I'm going to deliver today was by him only. So I owe a lot, to him as well. So today what I'm just going to do is, since this is a kind of an educative webinar, I'll just try to start with the basics of whole exome sequencing. What are the pitfalls? What are the challenges? And why and wherewithal this exome sequencing could be used. And we'll also slightly, talk about some of the aspects of exome sequencing studies that could be that could have been missed, but from a serendipitous study, we have discovered some kind of non-coding RNAs from exomes as well. So what is that? And I'll just slightly and slowly go in a nutshell. If you think that I'm going fast, maybe you can just drop a message to Avinash or in a public forum. So please feel free to stop me. Okay. So I come from Birla Institute of Scientific Research. So the next couple of slides is a quick introduction about BISR. So if you could see this, there's something called Moore's law. So Moore's law states that, from the computational perspective, the speed of the computing goes doubled, even as the size of or the need for what do you call a computational space also gets doubled with the cost coming down to a half. So the same applies to the genome sequencing as well. So this is what we call it as a sequencing economics. So you may say that the informatics costs keep dropping almost 2 times, with sequencing costs dropping almost 5 times a year. So that has been the case with several sequencing initiatives. And this part of the talk was done by my PhD fellow, whom I'm co-supervising, Sonal Gupta, and with my clinical collaborator Praveen Mathur and Krishna Mohan Medichella, who is my mentor as well. So we at Birla Institute of Scientific Research, we are based in Jaipur. Ours is a not-for-profit research institute, and it is run by the current chairman C.K. Birla, Chandrakant Birla, who has the biggest conglomerate of industries in the form of Hindustan Industries, and that office is based in Kolkata. Our academic sister concern is Birla Institute of Technology, based in Ranchi. And in fact, we are no way connected to BITS Pilani. Our academic institute is Birla Institute of Technology. And in Jaipur, as you could see, this is an iconic tower that you could see. We have a state-of-the-art laboratory facility. We have got 2 major departments. One is biotechnology and bioinformatics. The other one is a remote sensing department. And as you could see from the pictures, we also have the biggest auditorium, a state-of-the-art planetarium, and a so-called science museum, which is shaping up, with a big change now. And our executive director is Dr. Punendu Ghosh, and our head R&D is Dr. Krishna Mohan. And we are a group of 14 scientists, a small group. And in biotechnology, we are a group of 8 scientists, and about 7 PhD fellows overall, and about 2 postdocs. So as we could see, whenever we talk about next generation sequencing, the realm of next generation sequencing, comes from the basic adage of bioinformatics. So bioinformatics is a tool, okay, whereas its sister concern, computational biology, is a discipline. And when we talk about bioinformatics and computational biology, certain subjects like systems biology comes into picture. That's where your omic front is driven through systems approach. So if you say bioinformatics as a tool, computational biology as a discipline, system as any kind of entity. When we say a computer system, it could be any computer system. So if we take a biological system in the form of a human organ system, so we've got several body parts in the form of lungs, tissues, and all those kind of things. So these are basically some of the major components that we have. Okay, and these tissues in turn make up of cells. Cells are made up of certain organelles. In an eukaryotic cell, there are about 17 organelles. Okay, and of course, by virtue of the genetic material sitting in these organelles, there are 3 genetic 3 organelles that harbors the genetic material. One is the nucleus, the other one is mitochondria, and of course, the other one is the chloroplast as well in case of plants. So in eukaryotes, when we have these organelles, these organelles play a very, very important role, just because these organelles are not present in the cells, because the genetic repertoire present in these organelles plays a very, very important role for understanding the interactions present in the system. So these organelles, if you take nucleus has got nucleolus, you've got a chromosomal DNA that is intertwined. And if we uncoil the DNA into several pieces, bits and pieces, a bit of a DNA harbors nothing but a genetic material in the form of a gene. And gene, transcripts RNA, DNA transcripts RNA, and RNA translates to protein. And in an eukaryotic cell, unlike bacteria, in eukaryote, you've got a lot of introns and exons. In case of humans, there's only 1.8% of the coding material, whereas the remaining remains so-called non-exonic. I wouldn't even still call it as intronic, but you might have some kind of function deciphered, because this was once at a point of time believed to be junk. But thanks to the next generation sequencing approaches, there have been many non-coding RNAs that are believed to play some primary functional role. So this primary functional role allows us to understand what are the mutations that are arising and falling in the environment. What are the proteins evoluting and devoluting? What are those particular mutations that have got iconic changes in the sequences? And with these particular changes, would any of the proteins keep interacting with each other or not? And with that interactions, are there any kind of strains adapting or devoluting in the environment or unfitting in the environment? So this is how the lost and found theories have really come into picture. And the best essence that we can observe is from the COVID times these days, because it is believed that the virus has evolved from the bats, from the horseshoe bats. So so you see the beauty of it. So this is all done from the systems approach, and yet we need something called next generation sequencing approaches to understand this. Okay, so so this is the take home message. So you have got organisms. Organisms constitute organs. Organs constitute organelles. Organelles constitute certain genetic material in some organelles, and that is believed to take us the backend. So this is something like, if you really understand this theory, this is just like several vehicles like cars having smaller particles called vehiculars that are interconnected to each other. Okay, so that is the very basic essence of connecting two dots. One is the systems approach, and the other one is the sequencing approach associated with the systems biology. So in this talk, what I'm just going to give you is, I'll just try to give you the very basics of next generation sequencing, how the libraries are prepared, why is it we need to go for the whole exome sequencing approach. Since I've been asked to give a talk on the exomes, I'll probably talk on the exomes. And the systems genomics research, how this whole exome sequencing research would really lead into that. That's how most of my talks would be. And of course, we'll also talk about the regulatory genomics approach, the pathway tools for metabolism. And of course, we'll be doing some of the cases that I'm talking about. We have been working on congenital pouch colon, which is a rare disease based in Rajasthan. And I'll just try to give some kind of a gist on how the annotation is done for the next generation sequencing approach. And this yellow one, and I'll just try to give, if we have time, and if I'm if Avad and Avinash allow me. Okay, yeah. So the what of ohm sequencing. So when you say ohm, OME, ohm is nothing but many, okay, or money. जब भी हमें किसी को हम पैसे वाले बोलते हैं, तो then ये बंदा ओमी है करके कहलाते हैं. So this is how ohm is. So when we say genome, the study of many number of genes is nothing but genomes, and the study is called genomics. Okay, like as proteomics. There are about 347 omics that are well known these days. Okay, right from genomics, proteomics, metabolomics, transcriptomics, and etc. and etc. So if you really say take a bit of DNA, which we eventually call it as a gene, as a gene. DNA harbors both intronic and exonic chunks, as you could see from the cursor over here. Okay, so the exon is something in yellow color, the intron is labeled in black color here. So now the question here is, exons code for coding chunks, that those are the real translated bodies. So they give rise to the phenotypic expression, they give rise to the protein, and that's how the phenotypes or subjects in question are known. What kind of translational entities are deciphered from that. What are the kind of diseases, if at all, if there are certain mutations that bring out a kind of a change. So all those kind of things we can probably understand from this. So if you really see this from the sequencing perspective, these DNA is broken down into several pieces and fragments, usually um, in several equal components. Okay, so we usually call them, based on the number of fragments that they make, and based on the sequencing chemistry, the instrument has got certain capabilities. So if you really take 150 base pair chemistry, it could be a paired end, which means the sequencing orientation or read is done from F to R, right from the forward to the entry prime end, and also from the reverse direction as well, which we call it as a forward read and as well as a reverse read. So that is how equal bits and pieces of fragments are done. But in order to make them a good identity, there are certain clamps. These clamps are nothing but the adapters and linkers. So this is all from the wet lab perspective, where they are hybridized like this. Okay, so these adapters are nothing but, again, a kind of DNA bases, DNA sequences that are clamped to each end of these particular fragments, so that the hybridization spectrum can be read in a proper way. And then they're target enriched, then they're sequenced, and then you get these A's, T's, C's, and G's. Okay. And now the most important question is, when the targets are to be enriched, there are various strategies. I'm sorry for this, changes in the slide, because of this quarantine. Although I went to the lab in between, I use Windows at home and Linux at in my lab, and somehow there has been a change. And thanks to Avod, who helped me with these things, and yet we find some disparities here. I'm sorry, for those letters that are in a zigzag way. But the take-home message here is that there are different ways enrichment is done. So usually these fragments are hybridized and ligated, and the machine reads those particular fragments and then sequences. Okay, and it all depends on the kind of sequences that we go for. There are various ways and approaches. So for example, here for the target enrichment, there could be the inversion probes, PCR probes. PCR probes are nothing but, you can take the entire amplicon size of say even 1000 kb region sometimes, and take those particular entire amplicon as one particular probe, and try to read it, go for several copies of that particular sequence, and then read A's, T's, C's, and G's. There could be molecular inversion probes as well, and these are something called, what we call it as ISCs, or in solution capture based approaches, or direct genome hybridization capture based methods. Those are also pretty well known, but those rely upon different types of sequencing chemistry, different types of sequencing platforms, and different kinds of approaches that we usually go for. Okay. So the well-known, of course, 85% of the users are Illumina users. These are the short paired sequencing chemistry, which means the size of the fragments that are really chopped off or cut off is maximum of 500 bases these days, and the minimum was at some point of time even it was 50. Now the maximum is 50. The best and or ideal paired end sequencing chemistry is about 150 base pairs. So usually if we say paired end, usually we take 150 base pairs of each particular fragment, and take those particular multiple copies of those particular fragments from the two orientations, and then sequence it. In case of a single end sequence, you go for only one read that is oriented from 5' to 3' direction. There is also another approach which we call as a mate pair chemistry. If the lab is rich, if you think that you want a greater precision, although the precision may not really be directly proportional to the identifying any mutations. Mate pair chemistry is only done when you have several replicates or several sequences that are juxtaposed. So mate pair chemistry is another one where several orientations of forward reads and several orientations of reverse reads are taken into picture. Whereas if you really take the other sequencing platforms like Roche, okay, solid was erstwhile, that has been now considered as PacBio along with Oxford Nanopore Technologies. These are all the long read sequencing chemistries. where several bases, several kilobases of sequences are really read, and they are really chopped and then fragmented for sequencing. And then comes the real bioinformatics analysis. So the data needs to be processed. So usually the sequencing, the next generation sequencing raw reads are usually available in FASTQ files, FASTQ, which we call it as the fast quality files. And in case of the Oxford Nanopore technologies, they're called as FAST5 files. There are also some other sequencing platforms like Fastest, which was erstwhile for Solid, for PacBio, or for some other things. There were also something called FastM files, okay, which we called as fast matrix files. But ideally, 97% of the files, the raw read files which are originating in the form of FASTQ raw reads. And these particular sequences read based on the entire sequencing capture, based on the amplicon size, based on the hybrid capture, based on the PCR probes that we have discussed earlier. Okay, so this is what I hope it is audible. I hope I'm not going fast. I just wanted to be sure. Avadh?
No, sir, it's fine.
Yeah. So then now comes your challenge. You have those particular sequences. Now, if you generate those FASTQ files, the challenge is how to go about now. You usually go for mapping those particular sequences to a reference, if you think that you want to go for a reference-based approach, or if you think that you want to assemble your genomes based on certain de novo heuristics. De novo heuristics is you assume that you don't have any reference for that. You start right from scratch. This is what we call it as a nibble hypothesis, where you start right from the beginning without any taking anything into consideration. But you consider that certain sequences are there, right from your scratch. And then the FASTQ files are to be converted into something called SAM, the sequence aligned mapping files. That's where you map either to the reference. Okay, the SAM file is pretty huge because it maps it to the reference. But to make it worth easy, you need to convert those SAM to BAM, which we call it as a binary alignment mapping. And in case of eukaryotes, because we're talking about exomes here, nevertheless, we usually talk about eukaryotes only. So the BAM, when you get the BAM file, the binary alignment mapping file, which is in the form of usually the binary sequences, that needs to be sorted because it's not really sorted as per the chromosome of that particular reference. Okay, so if you take say Drosophila or human or anything, that needs to be sorted as per that particular chromosomal position, like chromosome 1 till the end. Okay, so that is also very very important. And then sorted BAM, then you get something called MPILE-up. You try to understand how these particular binary aligned mapping files are oriented across different directions and piled up, so that you'll not miss any kind of a stack overlay, which means those particular fragments that you have earlier split up, they are usually piled up one on the other. So when you pile up, there might be a stacking overlays, which means some of the bases in between might be given a miss. So those are considered in this particular direction. And then the most important thing in case of whole exome sequencing is calling the mutations, which we call it as a variant discovery. Okay, so for that you have several tools, like several online and open source tools like Anover, VT, WireScan, and several other tools. And then you map those particular mutations that you obtain to the publicly available databases that are there, which are in the form of Exact, the Genomate, the CADD, the GERP, and several things. But the most important thing is here, what I put up here. These are the different 1,000 genome databases and some variant predictors, the SNPF, the SIFT, several other databases that we really need to map it. The most important thing in exome sequencing is evaluating those particular variants. And then how do you evaluate? I'll come to that particular point later. So there are well-known pipelines. So there are well-known established pipelines that are commercial use and as well as open source use. The commercial platforms are present here at the extreme right hand, like the Partech, the Interpretomics, the Keagen CLC Bio. There are also, the star pipelines, that probably Bionivid and others, they use it. And the well-known pipeline, about 71% of the users use something called well-established benchmark pipeline, which we call it as a GATK, the Genome Analysis Toolkit, especially for inferring the exomes. But there are also other tools like SIGMOL, OPEX, Impact, and other tools. And there are also other platforms, online platforms like Galaxy, the user galaxy.org, which you probably well know. There are also other tools like Tavexa and other tools as well, where you can simply give your fast Q files and it will automatically generate the variants for your files. Okay. But the most important thing, the most important take-home message here is you need to reach consensus. Okay. So consensus is very, very important. And of course, once you reach the consensus, the most important stuff here is at the end of the day, you will get a final file, which we call it as a VCF, which is a variable, the variant calling format file. Okay, this particular file will define you what are those particular variants that are specific to your allele frequencies for your considered genotypes. Okay, so this is how you need to use it. There are different tools. And I think these particular tools are all well established. But as I tell you, as a systems geneticist or bioinformaticist, I would request everyone to tweak all the tools, which means there are different parameters for every tools, every open source tools that are developed. And those particular tools need some enhanced developments. There are certain changes, and every time you need to make it. You can tweak your tools, you can benchmark it. Maybe you need to use 2 or 3 other tools for every point in question, and then benchmark them. Okay, and then ideally reach consensus. That is the beauty of it, because it's all open source tools. It's all developed based on different ideas and hypotheses. So you need to come out with that. So we have also benchmarked our own tool, thanks to Narendra, who was my graduate student. He's in Oxford University right now. So this is, where we feel proud in saying that this is the highly accessed tools with about 20,000 downloads now. And our pipeline is well established, and this is from BioProtocol, which is a Stanford University-based pipeline. Okay, so we have got a pipeline which we have developed at BISR. You simply give your FASTQ file, and it will automatically generate the variants at the end and give you a list of those particular variants in your given population. Okay, this protocol can be accessed over here. So now the question here is, you get something called FASTQ file. Like the way you do in the lab, whenever you isolate small molecules, RNA, DNA, or say proteins or whatever, you need to check the quality of it, whether or not they are contaminated or not. In a similar way, here as well, you need to cross-check what are the readable, mappable reads in that particular sequence. Is it like the entire libraries are done properly, they're chopped off properly, whether or not the entire exons are chopped off properly, or any intron exon splice boundaries have really come into picture. Okay, or any adapters by chance might have crept in. Okay, whether or not the intended targets are covered, or whether or not the yield depth is quite good or not. So these are all various parameters that one has to really look into whenever we consider it. I always give an example of example of one thing. So imagine you parachuting or paragliding, say 10 kilometers above your place. So imagine, say you're in Bangalore, say 10 kilometers above say Bangalore, you are paragliding. Okay, and you don't see anything, but you come down 5 kilometers, probably you might be seeing some outskirts of say Bangalore or something like that. You come down, you jot down to about 2 kilometers, you could probably see the entire gray areas of Bangalore. And you come down, say 1 kilometer above above the earth, you could definitely see where Bionivid office is, probably in Electronic City or somewhere. Okay, and then you jot down, then you paraglide to your exact destination at somewhere at 500 meters above your sea level, above your earth, and that's where you can really exactly come into your destination. So this is what we call it as the depth of the coverage in sequencing terms. So where exactly, how many number of times your sequences have been read. So if you say that particular fragment that are being chopped off right from your sequencing chemistry entities. So if the depth of coverage is say 80, which means those n number of times that particular fragment has been sequenced, and the intended probes have been set for the targets, which means the depth is high for that particular sequence, which means it has covered innumerable proportions, so that the chance of identifying mutations is very high. Okay, so this is what we call it as this thing. And of course A, C's, T's and G's, which we call it as a GC percent. Usually in eukaryotes, the ideal GC percent is anywhere between 48 to 52%. Okay, that should be met. In some cases, in metagenomes, usually it comes down to about 38 to 44%, 45%. Okay, but for exomes, ideally the GC percent should always be covered. Now this is always a very important thing. And apart from that, for a whole exome sequencing, usually these days, earlier, there used to be only 30x or 40x. In case of the 1,000 genome databases, when the whole genome sequencing was launched, the coverage was only 2x, 3x, and 4x, which means only 4 times accurate depth of that particular fragment, that was chopped off, was sequenced. But now you see, even in case of exomes, even sometimes 500x doesn't make any sense, as long as you sequence or prepare your libraries absolutely good and ensure that your intended targets are quite good, or quite reliable. Okay, so there's something called interquartile range as well. So which means if you really see the mappable reads, which means the reads that are obtained from the fragments that are chopped like this. This is forward read 1, and this is reverse read. So what is the consensus between these two, where my arrow mark is? This is what we call it as an interquartile range. Okay, that defines whether or not your sequencing is really done properly or not. Sometimes the adapters are also would come into picture. In those occasions you may have to trim the reads. Okay, this is what we call it as trimming the reads, and that can be done using cut adapt or some tools like Trimomatic and all those kind of things. So in a nutshell, the take home message here is that you get sequences for your reliable targets. The most important take home message here is that you need to check the quality of your sequences. That is done using certain tools like multi-QC or fast QC. Okay, fast quality check, and those particular tools will tell you whether or not there is a good quality score. Quality score is usually measured by base calling approaches, which we call it as Fred tool. Okay, usually the Fred tool above 25 is considered optimally good. Okay, and if the quality is less than 25, it is considered mediocre or bad. Okay, so that is about very basics of next generation sequencing and all. But sometimes you might really wonder, there's another word that people often use, something called clinical exome sequencing. So when we say whole exome sequencing, you're taking the entire 3.2 GB of the haploid data set. Okay, taking all say 1.8 GB of it, so which comes to about 28 KB. That particular 28 MB, sorry, that that minimum is 28 MB and the maximum is about 39 MB. That particular entire region is really considered your entire exonic chunks, taking up all the exonic chunks across the human genomes. Okay, and then many a time you need not really go for identifying or going for exome sequencing of several patients taking the entire whole exome into consideration, whole exome into capture. Okay, probably you might really want to go for certain desirable phenotypes. So for example, in case of some diseases, probably you know what are those particular um, genes responsible for those particular diseases. So maybe you might really want to target only those exons or those particular genes from those particular areas only. So that is what we call it as a clinical exome sequencing. In a clinical exome sequencing, the size would be small. It doesn't necessarily mean that you would only chop off only the desirable genes here, but you would also look into the entire exome in proportion, but you would also take the sequences in a very nutshell, where this methodology is really followed. So you share the DNA, you prepare the libraries, as I told you, go for target enrichment, you go for generating the clusters, you sequence it, and all these things would really take somewhere between 20 days. Okay, this is really the time frame. So you might ask me, Prash, why exome sequencing, when we can go for whole genome sequencing? I think there are two answers for this. One is when exome sequencing came into picture, it was really believed that only the genetic variation was believed to be best seen only in the exonic chunks, only in the coding chunks, because as a result, you will only get the mutations that are seen only in those particular coding chunks, because that's where your phenotypic variables are seen. But as the whole genome sequencing of several genomes came into picture, right from the 2001 whole genome sequencing of 1,000 genome projects or whatever, it was believed that the role of non-coding RNAs also evolved. Okay, so in 2011, it was believed that the non-coding RNAs play a very important role. Until then, microRNAs were absolutely playing a huge role. But it was also believed that there were also long non-coding RNAs coming from the intronic chunks that are believed to play a very important role. So, so there's always, a question for researchers what to do, or what should be your, priorities, is it the whole genome sequencing or whole exome sequencing? And of course, the cost would always be variable. In case of whole genome sequencing, it would cost pretty high. In case of eukaryotic sequence, anywhere between 100,000 to say 500,000 per sample in case of whole genome sequencing. Whereas for whole exome sequencing, for an indefinite size of say a minimum 110x coverage, it would cost anywhere between 20,000 to 30,000, depending on different different companies. So the question here is, in case of whole genome sequencing, you're taking the both exonic and as well as intronic chunks, whereas in whole exome sequencing, you're taking only the exons. And of course, this is because you're only checking, from the diagnostic perspective, or maybe therapeutic perspective, or maybe these days, both, therapeutic and diagnostic perspective in the form of, theragnostic, perspective. So it serves as a powerful tool, and overall, at least in humans, there are more than 200 genes that are well known across, different types of, populations, different types of subpopulation, be it Indian, African American, Caucasian, Han Chinese, Australian, or whatever. Okay, and the only fallacy, of course, with whole exome sequencing is, as I told you, because the non-coding RNAs are seen in the intronic chunks, you might really miss identifying the genetic variations seen in the non-coding RNAs. Whereas in exome sequencing, you tend to get a lot of false positives. And again, the false positives is you don't know whether or not the non-coding RNA that you really identify is really a pseudogene or not. Again, you need a manual annotation for that. And most importantly, the depth of the coverage may not really be met. So for an ideal 20x, or even for an ideal 5x, or even for an ideal 10x, then you have to shell out a lot of money, because the gigs are bigger. The size of your entire genome is quite bigger. Okay, so for 3.2 GB haploid set, in case of a human, this thing, you are almost spending almost spending about 120,000 just for a depth of coverage of say 30x or 35x. Okay, so But now there are challenges because whole exome sequencing, the many sequencing efforts have really come into picture. People are really going for whole genome sequencing efforts these days. We'll also discuss, why. So what might be missing from exomes? So you get a lot of exons of genes. You might miss a lot of non-genic regulatory elements that are essentially the non-coding RNAs. Some of the structural variants, even the mitochondrial DNA is given a miss. There might be pseudogenes or non-coding RNAs that might essentially be coding, because it is believed that the non-coding RNAs also have some coding potential. But the good thing here is it costs only 1/7 of what whole genome sequencing costs. Okay, it's a fairly good price. You'll get a good diagnostic discovery phase for this, where you can identify the causal mutations. Um, and of course, the fallacy again here in case of whole exome sequencing is there is always a limitation for that. In either case, in either whole genome sequence, whole transcriptome, or whole exome sequencing at any given point of time, the ideal challenge is to identify what are the types of mutations that you have. Okay, the mutations could be benign, well-defined, malignant, the disease associated, the causal or bona fide mutations. The causal or bona fide or disease associated mutations are more pathogenic or likely to be, pathogenic mutations. Okay, so this is how we really need to look into picture. So in case of whole transcriptome shotgun sequencing or RNA-seq, the seed material is RNA. So you isolate RNA from your sample, go for second strand synthesis, making it into cDNA, and then that cDNA is really tested for gene expression. The transcripts are checked for relative expression across different samples. So there might be a normal sample, which we call it as a control, and there might be an affected sample. In case of plants, for whole transcriptome sequencing, it could be a treated sample or an untreated sample as well. Okay, so this is a bit out of question at this point of time. So in case of exomes, in a nutshell, in the genome you have about 4 bases, so which means about 3.2 billion bases in total per cell, and they're absolutely identical for a haploid data set. And in proteome, when we say proteome, these code for about 20 amino acids, which correspond to about 60,000 genes in case of humans, about 200,000 proteins. And these 2,000 proteins make up different peptide fragments in the form of enzymes and metabolites and all those kind of things. And these are relatively making up, several expressions in the form of, about 200,000 pathways. And roughly there are about 1,000 pathways in an ideal cell. And that's where, your differential expression, really comes into picture. And for this, there are several tools as seen here. Okay. At the end of the day, what really matters here is, for a whole exome sequencing, whether or not you're ideally identifying any mutations per se or not. Okay, so if you're identifying mutations from a sample for that particular sample for a disease condition, what are those particular mutations that are present in an affected sample when compared to a healthy or an unaffected sample? That is where we could understand. Okay, so this is how whole exomes could really bring up something called the cure me now immediacy, which means many people who don't know what are the kind of mutations present in those particular samples would really go for clinical exomes or whole exome sequencing. That's because these are better for theragnostic approaches. And this is how you can even develop a reconstruction of different pathways. That's because if there is a mutation, that particular change in mutation might result in a different phenotype or a different protein moiety. And if there is a change in that particular protein moiety, that particular protein might interact with another protein in a different way, in a different cell, in different conditions. And if they're really interacting in different conditions like that, then the pathways might change. So that's where you can reconstruct your pathways. Okay, so the next 20 minutes of my talk, probably I'll talk about our case studies on the rare diseases. I've had about seven and a half years of my postdoctoral experience, working across different labs. which I learned in Denmark, US, Estonia, South Africa, and Japan. That really helped me a lot, to conquer certain thoughts on how, the next generation sequencing approaches, would really come into picture. But here, when I returned back to India, our clinical collaborator and Dr. Praveen Mathur and Dr. Krishnamohan, our head, they had this congenital pouch colon, which is a rare disease, which is often seen in Udaipur and Udaipur district, and also some parts of Pakistan, some parts of Punjab. There were 1 or 2 reported cases in Germany and Denmark as well, where the child is born with a dilated pouch, and with the dilation, it brings out a lot of urinal retraction, which means the child cannot pass on urine properly, and it's really excreted all through, and it's in a very messy situation. And Praveen Mathur and their group, they have identified a lot of types of congenital pouch colon, as you could see here. The child needs to be operated probably within 3 months ideally, but there are also children who needs to be operated at a later stage as well. So what we did was we identified causal mutations specific to congenital pouch colon. So this is again a simplest whole exome sequencing approach. You can see our paper here, Mathur et al. 2018 in Scientific Reports. So that was a very basic palette study, but now we are coming up with something called trio analysis. So it's a rare disease. It's native to only fewer northwestern states of India. We don't know the possible reason. The possible reason could be because of poor nutrition, or maybe some people eating a typical unbaked bread, or typical jowar kind of bread, or it could also be believed to be because of different contagions present in the water, because of the fluoride content in the water, or because of the air quality where the child is really born with this. So the last 20 years, ever since Dr. Praveen Mathur has been doing all these kind of surgeries, he's also a bit clueless why these particular things have really come into the picture. But we have identified certain mutations. Those mutations and genes are better captured in this particular paper. But what I'm trying to tell at this point of time here is we did about 64 exomes, which means there are about 18 probands, 18 children's sample we have taken, along with their family sample as well, father, mother, and as well as sibling. So we wanted to see what are those mutations present in the child that are absent in father or mother. So this is basically, ideally we call them as de novo mutations, which means if there are mutations present only in the affected child but absent in father and mother, then that's how we know the take home message for us. And our paper is currently under preparation, the trio analysis. The trio here is the affected child, which we call it as a proband in clinical terms, the father and mother. Of course, we also go for something called sibling. We also check whether or not a sibling might also harbor certain mutations, if at all if mutations are carried forward, say from mother or father. Okay, but in either case, all the 16 probands, in fact we had 18 probands, but for other 2 probands we didn't have father, mother, and sibling samples with us. At least for the 16 probands, none of those particular parents have got any carriers from their frontline conditions. Okay, so we were a bit curious, and our analysis really giving it to different pathways, different colon or colorectal related pathways. Okay, and I'm also working on cancer prostate these days. We're also working discovering cancer prostate exomes from Indian subpopulation. We have got a lot of links associated with some of the mutations associated with colon as well, or rectum as well. Okay, so basically what I'm just trying to tell you during the next few slides is that we have something called whole exome sequencing. So ideally in a whole exome sequencing, you try to only chop off the exons. So there's no question of introns here. Okay, so there's no question of splice junctions or even the intronic chunks chopped off as well. But there might be a possibility sometimes because the capture methods in case of the Agilent capture based methods like V5 plus 5' UTR capture based methods, or the star aligners, the BioStar or other capture based methods that are publicly available. These capture based methods ideally work in such a way that you can even chop off ideally the intronic chunks to a specificity. Okay, so ideally one fine day what we tried to do was we asked because I work on long non-coding RNAs as well, that's my interest. We try to check whether or not any of these particular sequences have got any long non-coding RNAs. And to our surprise, when we did the Smith-Waterman algorithm, which is ideally a kind of a blast, from our exomes, we found long non-coding RNAs as well. So which means ideally, when the library was prepared, there were some intergenic non-coding RNAs also spliced, which means there are some intronic splice junctions that are present in between two exons that have also been captured when the sequencing was done. And this was ideally seen across several samples. And then we thought, oh yeah, it was a very hurrah moment, maybe it's a Nobel Prize award-winning moment. We found non-coding RNAs from exome captures. We thought hurrah, hurrah, and we did several approaches. Of course, as a systems geneticist, we do several approaches. We get it checked twice or thrice. I asked my PhD fellow Sonal, to also do that. We did some experiments as well to check that, and this is the basically this particular story. So basically, what I'm just trying to tell you here is, exomes doesn't necessarily mean that you need to capture only the exonic chunks. Okay, so you might also end up getting the intronic chunks as well, thanks to the non-coding sequencing chemistry parameters that we can really look into the picture, and thanks to the sequencing librarians as well. We might even ask the sequencing librarian, someone like Awad, to help us, Awad, please try to capture on these particular regions as well. So it's basically a kind of a targeted amplicon sequencing. Okay, and this is what we did. We isolated that long non-coding RNA which we identified from that, and we used something called microscale thermophoresis from Oxford Nanopore Technologies. From Nanotemper Technologies, I apologize. Okay, a good friend of mine, Shivram, based in Bangalore, he did this. He's the head of it. So what basically does is it checks whether or not two small molecules that are allocated in your append-off has got highest chance to interact physically or not. So it's basically a kind of a pull-down assay. It's basically simplest Western blot or a co-immunoprecipitation or something like a histohybrid screen. Okay. So this is what we did, and we tried to ask if there are any mutations seen in the non-coding RNAs as well. Okay, so this is a very important thing we did, and we amplified the product, and we have raised primers for that non-coding RNA. The non-coding RNA is something called non-HSAT, which means non-Homo sapiens, non-coding Homo sapiens transcript ID number 008982, and that was the long non-coding RNA we found. And we checked it using Experian, the nanodrop thing. Ideally, as you could see, the peaks were absolutely jubilant. They were absolutely good. We got the RNA tissue samples analyzed, and we got the quality controls tested as well. And then we also checked what was that particular ideal protein to which that long non-coding RNA was interacting, and ideally that is KIF13A protein. Okay. So the food for thought here is, whenever we talk about exomes, ideally you don't talk about mutations only. Even if you were to talk about mutations, you would only check whether or not the mutations are present in your sample that are absent in, say, father or mother, if at all, if you're going for trio analysis. Or even you might want to see whether or not those particular mutations are specific to your geographically niche area of subpopulation as well. Maybe you might be belonging to a very rare tribe in India where your sample has got certain mutations which are not even seen in the other components of Indian subpopulation as well, or maybe in Caucasian, or maybe in some other regions as well. So that's where your whole exome sequencing capture technology would really come into picture. But from exomes also you can definitely go for non-coding RNAs. Ours was ideally the first lab, but publication wise we were a bit delayed because we were writing the paper, and we published it in Biomolecules with an impact factor of 6.1. Ideally the first paper came in PNAS. So probably we missed the Nobel Prize. This is a very hurrah moment for us, I would say. And thanks to the works of Sonal, who has done this particular work. She just submitted a thesis based on this as well. So basically the problem we asked was, does non-coding RNAs have the mutational spectrum from exomes? We asked, how do we define that challenge? And then we ideally looked whether or not those non-coding RNAs ideally have got any regulatory role, any transcription factor binding sites, or something like DNA's hypersensitivity sites or something like that. Okay, so this is what we asked. Other than Avinash, how much time do I have?
Sir, you can continue. No issue.
Yeah, so I'll take, I've got about 7 slides, so I'll not bore you much. So what I'm just trying to tell you the next is, I'm not sure what's the target audience here, but I'm not sure if someone knows a very important term called linkage disequilibrium. This was my postdoc work that I carried out in 2014, which I wanted to share with everyone here, because this is one of the well-known works that I keep discussing every time. There's something called linkage disequilibrium. So in linkage disequilibrium, you get a lot of SNPs that are present in smaller range, smaller chromosomal sections of your ideal context, ideal loci, and you don't know what is a causal SNP, from those particular regions. So this is what we call it as a linkage disequilibrium. And slightly, if you really take this analogy into the genes, this is what we call it as pleiotropy. Pleiotropy is several number of genes have got unequal associations with several diseases as well. Okay, like BRCA1 and BRCA2. It's not only the causal for breast cancer, but also for prostate cancer in male. Okay, so that is very defining problem. So what we try to do here is, I mean, the reason why I'm telling you, you might ask me, what is this related with exome sequencing or next generation sequencing? The reason why I'm just trying to tell you is, here, this is a region, this is a high density map where we have a desired phenotype for obesity here. Okay, and this particular region is also associated with something called clinical mastitis, which is often seen in cows, and also in female genitalia as well, also in breasts, and also the region where the region is pretty bulged, and you don't know what is the disease from that, and the entire region is really affected with a bulgy skin with red dots and all those kind of region. So this particular region has got a lot of mutations. You don't know what is a disease. So what we did was, we tried to prioritize SNPs. How do we know whether or not this particular SNP coming from this particular chunk, be it exonic or intronic chunk, is really causing a disease? Okay, so we checked, we walked with something what we call it as genome walking. We go for 5' UTR region of that particular SNP. We go further upstream of that particular region and see if there are any functional elements, okay, like transcription factor binding sites, and if there are any constrained elements, like any evolutionary conserved regions, if there are any regulatory elements, which means any non-coding RNAs present somewhere upstream of that particular SNP, okay, we identify. And then what we do is, we take those particular SNPs across different elements. So each of these pies are different elements. One is a constrained element, the SNP coming from constrained element. this particular pi is coming from, say, the functional characterization, and this is, say, from something else. So what we try to do is we try to prioritize the SNPs coming from different regions. So as you could see here, there is an orange SNP here. Okay, the round small ball-like thing is a SNP across different pies. So if ideally a SNP is present across different pies like this, so which means in this case, a SNP is present here in the constrained element, in the functional characterization, and also in the regulatory elements, and also in the pathways. So if this SNP is associated across those 4 things, then we call this particular SNP as a causal SNP. Okay, so this is a prioritization approach that we did for a disease phenotype of our interest, and this data is really coming from exomes as well. Okay, so that's how you can even characterize the SNPs from the differentially expressed genes or differentially expressed long non-coding RNAs. We can do ideally different things. So this is basically a kind of a genome walk theory, where ideally you have a SNP somewhere here, somewhere a SNP here, and you walk upstream of that particular gene and see if you find any regulatory elements or constrained elements or functional annotation. And if you find it, then that particular SNP is associated, say, across all those 4 pies, then that particular SNP might be associated with a disease. Okay, and this is how we try to bring up a solution. Okay, so so the overall conclusion here is we have identified certain SNPs that are causal for congenital pouch pullen. I didn't discuss the trio analysis. The trio analysis for a rare disease is very unique. There are only 9 or 10 papers for trio analysis that has been done for rare diseases, not many. And for bigger scale, where we already have about 18 samples, is quite a big one. Our paper is coming up in a very good impact factor journal, which will be coming up very soon. So whole exomes have been a preferential method for inferring causality. And we also have prioritized SNPs if those particular SNPs are coming from different chunks. And that thing is what we call it as a 3-point classification strategy: the regulatory elements, of functional elements, and basically the constrained elements. Okay, and there are also different pathways that we could really map to. And whenever we talk about exomes, ideally we don't necessarily talk about the exonic chunks. We could also say the non-coding RNAs as well, because we can ideally identify the non-coding RNAs, especially the long non-coding RNAs. So you should probably thank the sequencing librarians, or have a good dream, overnight, and think of your particular candidates, especially the non-coding RNAs, which ideally could really allow you to identify them as good markers. And of course, if someone like me, I would probably look into something called long non-coding RNA protein interactions. So in about 1 hour from now, we have this panel discussion. You're most welcome. You can click at bioclues.org/events. We've got eminent speakers on the board. We are trying to discuss where are we heading for diagnostics of COVID-19. I'm associated with BioClues as well. I'm the founder of BioClues, which is India's largest bioinformatics society here. So I would like to thank Avadh, Avinash, and Rohit, for their kind invitation. I'm grateful to them for their wonderful time and energy. And this is my group page. You can click here and you can see this. And we thank, SM and KM has funded this particular project, but I got, funding from a brain pan for prostate cancer, and also served EST for vitamin K pathways, and also a couple of mini grants as co-investigator for DBT and BTSNet. So the whole work was done by Sonal Gupta. Praveen Mathuri was a clinical collaborator. Of course, KM is our head. And Saji Menon and Shivram, they were the ones who worked for our, the microscale metamorphosis, which I was talking about. Yeah. So thank you so much. I'll just wait for your questions and answers. Sorry for my answers. Yeah.
Thank you so much, sir. Thank you for your talk. There are a few questions that have come up. First one is from Ms. Hemangini Naik.
Yeah.
I hope I'm audible, sir. Yeah.
Yeah. Yeah, please, Avinash.
Okay, so her question is there any possibility that the adapter sequences or any contaminants appear in the FASTQ files?
Yeah, very much. You'll definitely find them. Many a time, even for example, even for the COVID-19 sequences, the assemblies that have been done, you find a long trail of sequences which you can clearly see that they're the adapter sequences which you really need to ignore them. So the assembly, needs to be done properly. Of course, the library preparation needs to be done properly. And even if there are adapters, that needs to be done, that would already be already have been truncated using the cut adapter trimmomatic tools that you would probably use. So many a time in whole exome sequencing, ideally you're really looking at only 30 MB region ideally, so you'll not find major pitfalls with respect to exome captures especially. But in case of whole genome sequencing or whole transcriptome, metagenome or meta transcriptomes, ideally you might find a lot of these gray areas where you need to carefully assemble those particular sequences. You definitely find them. Yeah.
Okay, Dr. Prashant, we have
Yeah.
with us, Dr. Roli.
Yeah. Dr. Prashanth, I have actually two questions from your presentation. So the first question is wherein you are explaining about the non-coding RNA, the link RNAs. So basically what I know that nowadays panels are also available wherein they give you a like non-coding RNA as well along with the exome sequencing. But in your case, the thing like you have done exome sequencing, you haven't used this panel, right? You would be getting a chunks of some intergenic regions, what you have detected, and you have detected it using BLAST. So my question is that chunk, we also, in our experience also, we get a lot of SNPs lying in the intergenic region, but we cannot trust them because the thing is like you have BLAST with a maybe a very smaller chunk of intergenic region, whatever you have. So how much we can rely on it that of course it is non-coding RNA.
So I mean, I'm not sure if you have seen the entire talk of mine. So what we do is we did something called Smith-Waterman algorithm. So Smith-Waterman, unlike BLAST, it's more sensitive. So it is it's just like, it's not a kind of a fast approximation based approach. So what it does, it ideally identifies whether or not there are exact matches across the length and breadth. And this was followed by something what we call it as a bidirectional best blast hits, which means now we take the target as a query, the erstwhile target as a query, and we take the erstwhile query as a target, and we check if we really get the same result in either ways. And this was followed by our exact isolation and checking those particular regions where we have amplified the long non-coding RNAs, and checked it using the microscale thermophores as well. So basically, you will not really identify what you call the long non-coding RNAs at every point of time. You would really find these long non-coding RNAs exactly at the intergenic chunks. Ideally, in our case, the hurrah moment for us was there was only about 2.3 kb region that was over-spliced or chunked from the intron-exon boundaries, which was chopped off from the sequencing library preparation. And ideally, long non-coding RNA would be more than 200 bases in length. And ideally, this long non-coding RNA which we have gotten was about 800 bases in length, which was well annotated, which was well curated. We also tried to isolate them from the lab from the same samples, which we got it. And then we also got it tested, with the amplicon-based approach, and also we tried to check it whether or not that long non-coding RNA would interact with the protein of interest, which is causal to CPC. So that is what we have done. Yeah.
Okay. And my another question is regarding your linkage analysis. So I think so you started from, you have identified SNPs, you scored it based on and you have grouped it, I think, and then
Right.
all that essentially as a diseased or not. I would like to know the upstream part of it, which I think you haven't mentioned in the presentation, how it was done.
Yeah, basically, these particular data sets are all from the high density maps of cattle, actually. The lab that I was working on, they established a lot of, the genotyping by sequencing based approaches, and also high density maps for cattle, pigs, and also, for some cases of the humans as well, some human obesity index. So the region which I was really showing where there's a high linkage disequilibrium is the chromosome 6 in cow, which is in case of, if you take the human analogy, it is chromosome 4 in humans, where the genes are between GC and NPFFR2. So these two genes are ideally responsible for causing, vitamin D deficiency. And in case of cows, these two genes also have something called, somatic cell count as a disease phenotype, which means it is also associated with a lack of, production of, good milk in case of, cow. In case of, cattle, I would say, not really cows, but it could even be associated with buffaloes as well. So once these high density maps are generated, you get a lot of data. These particular data are coming across, different sequences and population. So this chromosome 6, ideally between GC and NPFFR2, it is about 23 kb region in size. So between this 23 kb region, there is only about 3.8 kb region where there is a high linkage disequilibrium. So which means out of 3.8 kb region, which is about 3,800 bases, there are as many as 2,400 SNPs. So which is almost 70% of the entire region where we call this as a linkage disequilibrium. So the challenge here is to identify which of these particular SNPs are causal, which of these particular mutations are causal. So what we try to do is we try to genome walk. We ideally jot down to one SNP each. So out of those 2,400 or 2,500 SNPs, we'll selectively choose one of those particular SNPs that are ideally considered as putative or bona fide or probable, probably associated with that particular SNP that various researchers have identified. Now we go upstream of that particular SNP somewhere, even we walk up to 2 or 3 kb region, and we see if there are any transcription factors associated with that particular gene of interest, which is showing a high influence on that particular SNP. And also if there are any enhancer elements, if there are any something like open chromatin elements, or even ideally some of those particular transcription factors which play a very important role in playing in giving an enrichment for that particular GC or any of those particular genes downstream. So ideally the take home message here is if the SNPs fall within the vicinity of these particular transcription factors, the regulatory elements, the enhancer elements, the constrained elements, or the functional moieties, basically that SNP has got the highest chance to be causal or pathogenic. And now the challenge here is these are all the high density map SNPs. So we cannot really trust from the basic bioinformatics sequences. So we need to experiment in the lab. Of course, these particular SNPs were done later in the lab, but my work was, to come up with a prioritization strategy, for the SNPs as well. You can find in a couple of publications related to that. Yeah.
Okay.
Yeah. Sir, Mr. Junaid has asked a question. He's asking, is it possible that whole exome sequencing will not find some mutations that are actually present in the DNA?
That's certainly a good question. In fact, I wouldn't say it will not find, but as we discussed, there's something called the average depth of coverage we usually talk about. So ideally, these days, the exomes are not to be trusted trusted because you might realize a lot of those particular mutations that do not come under a good depth. Although for the clinical exomes for certain diseases, the depth of minimum of 5 is considered in many cases. Even there were cases where ideal mutation wouldn't even fall under that basic depth of coverage of 5. So basically, that particular mutation in that particular disease phenotype might really be associated with some of those subpopulation centric genotypes that are already published in GenBank, like the EXACT, the DBSNP, the CleanVar, the GERD, the CAD, the Genomate, which I was all really talking about across different subpopulation. And then you cross-check whether or not those particular mutations are in principle with respect to the minor allele frequencies. Okay, so usually minor allele frequency of 0.05, which is a 5% chance of that particular allele sitting in that particular region, is really causal to that particular disease phenotype. In case of rare diseases, the minor allele frequencies of 0.01 needs to be done. So if you really don't find those particular mutations, then ideally you'll not consider the particular mutation at all. So there are two theories. One here is, first thing is whether or not your quality check was done properly right from the beginning. That answers whether or not you really find that particular mutation at all. So that's one of the reasons why you probably need a stringent variant calling approach. You need to reach consensus. You need to call the mutations across 2 or 3 tools. So today, if I really run a pipeline using a GATK, which is a well-known pipeline where 71% of the users use it, and if I run my pipeline, you would definitely ideally find 1 or 2% change. But among those 1 or 2% change, you might really miss your mutation of interest. Okay. So the take-home message here is reach consensus in order not to miss those particular mutations. Yeah.
Okay, so we have 2 questions from Ms. Ankita.
Yeah.
Just a second. Yeah. Her first question is, what all precautions and steps at wet lab level should be taken for achieving a good enrichment of the probe used?
Yeah. I think the best thing is you always need to allocate your samples very good. An ideal wet lab biologist would repeat the experiments at least twice or thrice, but since the libraries are pretty costly, you don't usually repeat it, but you usually check a quality control where the wet lab biologists take preferential occasions. So they already have those well-known library prep approaches. Those channels are already available. Those are all benchmarked. I don't think any wet lab person would really give it a miss, because what all they really need to do is add those particular chunks of those particular units from time to time, like buffers and all those adapters and all those particular things in real time, then go for enrichment. So the enrichment tags you definitely get ideally. So probably that's where you need to trust your wet lab person or your service providers. Yeah.
Our second question is, what important things to keep in mind while analyzing the VCF from diploid organisms as compared to when we are analyzing that VCF file coming from haploid ones?
There's no question of ploidy in terms of analyzing the VCF files. Probably you would only look into something called heterozygosity, okay? Because if whether or not your allele is really sitting across 2 different strands, or your particular mutation is sitting across 2 different strands, and you would only check whether or not your mutation is really benign, if it were to be benign, if it's not causal, whether or not that benign mutation is also just because of something called homozygosity. Okay, so the ploidy level parameters can only be checked not at the VCF file, but probably the quality check that you need to ascertain comes from your BAM. Okay, not certainly at the VCF files. I hope that answers my question, your question. Yeah.
Okay, so Mr. Rajesh is asking a very generic question. His query is regarding the use of R programming in NGS data analysis.
Yeah, there are a good number of tools in R as well, like NGZ, which is a well-known tool for R, although I never used. That R program could also, I don't think, R is suddenly the need of the hour unless and until you really want to have certain dynamic plots. For dynamic plots, you could use something called clustwiz, which is an algorithm in R, where you can jot down certain maps like the principal component analysis or the heat maps that you generate, taking the exonic SNPs or intronic SNPs. Yeah.
I think that's pretty much it, sir. We have covered all the queries.
All right. Thank you so much, all of you, for your patient hearing. Yeah.
Thank you, sir.
Yeah.
Okay.
Yeah. Bye. Yeah.
Bye.
Thank you so much. Yeah.
Thank you so much, sir. Thank you for your time. Thank you for sharing your insights on the topic with all the participants. I would also like to thank all the participants for patiently sitting, listening to the talk and sharing their set of queries with us. So thank you once again. Thank you, everyone. We'll be ending the meeting session now. Thank you so much.
Yeah. Thank you. Bye. Yeah.